for the invitation, which I gave from John originally. It's great to be here, great to see you all, some certainly familiar names. I should introduce myself, just so you know who's talking to you. So I did my medical training and PhD at UCL, did a PhD in genetic epidemiology, which was mostly about Mendelian randomization and the use of genetics for evaluating drug targets in cardiometabolic disease. Spent some time as an academic clinical trainee in the NHS based at UCL and Imperial, and then moved into industry, and have been working sort of straddling genetics, genomics, and early phase drug discovery and development for the last 5 years or so. At Silence, where I've been for almost 3 years, I work on a combination of our clinical development programs. I'll introduce one of those to you today. And I also run our translational genomics group, which John helped to establish a couple of years ago. And I can tell you a bit more about that as well. But please do shout out with questions or comments whenever you like. I'm not sure if there's an easy way of doing that through a chat or whether you just want to yell. I'm happy with whatever you like. So let's get started. Try and make this advanced. So we're a publicly traded company, so we have to put this up. So anything I say is not intended to influence any investment decisions. So Silence Therapeutics, just to introduce the company so you know where all this is coming from, is a fairly small UK-based biotech. We're about 125 people based in the UK, in mostly around London, in Berlin, where our lab site is, and in New Jersey, New York. And we specialize in the discovery and development of siRNA drugs, so short interfering RNA drugs. The company's been going for almost 20 years and has seen a major resurgence in the last 5 years or so, where we've got 3 active clinical programs and others on the way, and some major partnerships with large pharma organizations. So the company is doing an awful lot, and the whole thesis of the company is currently based on this this siRNA technology, which has got a fancy name. It's called the GOLD platform, but it boils down to technology that uses a double-stranded siRNA molecule to downregulate expression of a particular gene, a target gene, in a tissue of interest. In the case of this platform, the tissue of interest is the liver, or to be more specific, the hepatocyte in the liver, and I'll come to how that all works in a moment. So our drug molecules are composed of three elements: this double-stranded siRNA molecule, then a linker bit, which is just sort of a bridge, a coupler, and then this ligand called GalNAc, which is N-acetyl galactosamine, which is a sugar, and it's the naturally occurring ligand of a receptor, which I'll show you again in a second, called ASGPR. And that receptor is the key to the delivery mechanism for these drugs to get them into the hepatocyte in the liver. So the really key bit of this is the siRNA molecule, but the way it gets to where it needs to do its business is through the linker and the GalNAc. So how does it do that business? This is a bit of a schematic of how this all works. So on the top right, you'll see those 3 elements of the drug molecule. They're delivered to the patient or to the trial volunteer subcutaneously, so small injection under the skin, fairly small volumes, and the drug, with its 3 parts intact, makes its way through the circulation to the liver, at which point the GalNAc, this sugar component, binds the receptor called the ASGPR, the asialoglycoprotein receptor, which is very highly expressed on the surface of the hepatocyte and in very few other places in the body. So there's this this major enrichment for this receptor on the surface of the hepatocyte, but very few other places. So by this sort of lock and key mechanism, the GalNAc is the key, the ASGPR is the lock. GalNAc has no other locks in which to fit. So it binds the ASGPR, and that then starts a process of intracellular transport. So the whole drug molecule is moved inside the cell, where it can start to have its pharmacological action. Once it's inside the cell, the GalNAc and linker split off, and the sRNA molecule then dissociates into a sense strand and an antisense strand. The antisense strand is designed to be effectively perfectly complementary to the sequence of the transcript of the target gene that we are seeking to downregulate. So the genes are transcribed into mRNA. mRNA exists as a range of transcripts that are eventually translated into peptides and proteins. By selecting the most appropriate transcript for a given gene, we can then determine the sequence of that transcript, generate the complementary sequence in the sRNA molecule, which is only 19 bases long. These are quite short, but we can still achieve perfect complementarity within 19 bases. That complementary 19 base sequence then binds its target sequence in the mRNA, in the transcript of our target gene, and induces a naturally occurring process within the cell called centers around this a protein complex called the RISC complex, and initiates degradation of that target mRNA that's been bound. So this is probably the key point of specificity of these types of drugs, that by exploiting Watson-Crick base pairing and this perfect complementarity of our drug molecule to its target sequence, we can make sure that we're hitting just the gene we think we're hitting. Now, of course, there are risks of off-target effects, and part of our design process for these molecules is to try and predict and measure possible off-target effects, but we can achieve pretty good specificity with these 19 molecules. The other neat bit about this is that the antisense strand, once it's marked the transcript for degradation through RISC, can then go around the circus again, mark another transcript for degradation, and keep the pharmacological effect going. So by keeping levels of the target transcript low, the consequence of that is that production of the gene's protein product is reduced or completely abrogated. And you'll see later when I show you some of the pharmacological results in animals and in people, that can last for several months. So that's the sort of nuts and bolts way that these drugs work. And I'll just pause there in case there are any questions about that, because it can be a bit fiddly. No? Okay, let's go on. So to put this in context of other drugs and why there's a lot of interest in this type of technology at the moment, obviously the massive explosion of mRNA-based vaccines has been a big boost to mRNA-based therapeutics in general, and I think the demonstration of these drugs in vaccine form, so the Moderna vaccine and the BioNTech Pfizer vaccine use similar technology. Demonstration of efficacy and most importantly safety have been a big boost to this modality. When you compare the success rates of sRNA-based drugs to drugs from other modalities, so small molecules, monoclonal antibodies, peptides, gene therapy, cell therapy, the success rates, at least so far, and I say so far because the sample is small, so there are fewer than a dozen approved sRNA drugs, whereas there are tens of thousands, probably not quite as many as that, but several thousand approved drugs in other modalities. So not completely apples to apples comparison, but nonetheless, based on the data we have already, the success rate of these types of drugs, phase 1, phase 2, and phase 3 is quite substantially higher than other modalities. And there are a number of reasons for that. Yes, it could just be small sample bias, and once the numbers even out, we'll see things even out. But I think there's certainly a safety advantage. We see very promising safety profiles with these drugs, a number of which have been approved. In fact, one was approved last night in the US for treatment of amyloidosis. But also because of that high specificity of targeting that I showed you just now, that boils down to the complementarity of the sRNA sequence for its target gene, you avoid a lot of the toxicity problems of small molecules, say, where their specificity can never get as high as that because you're relying on more traditional chemistry of ligand receptor binding and small molecule to protein binding, which is a lot muckier. So I think there probably is something in this, and it's driven by these important features of the technology. So let's look at a couple of, or 3 examples. One that may well have come across your headlines in the last year or so is inclisiran. So this was approved at the end of 2020 in Europe, earlier this year in the US. And this is a drug that uses almost identical technology to the drugs that we are working on at Silence. Its target is PCSK9, which those of you working in cardiovascular genetics will be very familiar with. This is a gene that's integral to the metabolism of cholesterol and LDL cholesterol in particular, and is already the target of 2 marketed drugs and others coming through as well. And if you look at the plot on the top left, you'll see what I mean about the duration of action and this recycling of the drug in the hepatocyte and having this long pharmacological action. So this is data from a phase 3 study published a couple of years ago. And you see there's dosing of inclisiran, subcutaneous dosing, as I described before, on day 90, then 6 months later and 6 months after that. And you see this very long duration of reduction in LDL cholesterol, which is on the y-axis. So barely any return towards baseline between doses. And that's the reason this technology, particularly for inclisiran, is being described as sort of vaccine-like, because the duration of action is so long. And inclisiran is approved for dosing at baseline, then at 3 months, then 6 monthly thereafter, so twice a year thereafter, which is unheard of for a therapeutic outside the vaccine world. It's not the only drug that's been approved, obviously, although it's the drug that's been studied in the greatest number of people. So about 11,000 or 12,000 people have been studied in published studies for inclisiran so far, and that underscores the observation I made just now about safety. So the safety signals from inclisiran have been pretty mild, mostly around skin reactions, so redness, a little bit of soreness at the injection sites, but so far no major safety concerns. Now, lowering LDL cholesterol is great for treating common disease, so cardiovascular disease, atherosclerotic disease, many tens of millions of people in the world affected. But a number of other drugs have been developed for rarer indications. So patisiran and givosiran, both sRNA drugs, and developed for much rarer diseases. So for amyloidosis in the case of patisiran, and porphyria in the case of givosiran. And I show these just to illustrate that the technology seems to have value in rare disease as well as common disease, and also we get this similar pharmacological effect with very long-lasting effects. So sRNA has been termed genomic medicine. I think not without good reason. I've taken to describing it as a a drug modality that directly modulates the relationship between the genome and disease in a way that other treatment modalities, small molecules, for example, they modulate that relationship, but less directly. They work further downstream at the sort of protein and further down part of this flow. But if you imagine this causal path from DNA sequence variation through transcription, through translation to proteins, complex phenotypes, then onto disease, we're acting pretty far upstream. The only further you could go upstream is with gene editing technologies, the CRISPR, for example, which is being done. So there are companies working on CRISPR-based gene editing for treating familial hyperlipidemias, for example. But for reversible therapies, gene silencing, RNA interference is about as far upstream as you can go. And many of you will be familiar with the data that are available in this world of research, which mean that investigating possible targets, modeling the anticipated consequences of modulating these targets, both positive and adverse, is very achievable. We have vast amounts of data, and many of you have been involved in generating those data on each of the steps along this causal pathway, starting at the sequence variation, starting with GWAS arrays, and now with whole genome and whole exome sequencing. We have very rich eQTL data from resources such as GTEx, but also others, and at Silence we've got partnerships with academic collaborators to generate new data, which has been great, so specific to some of our research needs. pQTL data, again, vast amounts of data coming through from Interval, UK Biobank, Decode, and elsewhere, and many more of the other omics in between, other XQTL data sets, and of course lots of information about the relationship between genetic variants and disease. So if we have a drug modality that can modulate gene expression, and we've got data on all of the bits of the causal pathway up and downstream of it, we can start to build quite interesting models of what might happen if you modulate the expression of a given gene, or even going down to a given transcript, which we can come back to at the end. So I thought I'd just give quite a neat example of this. So lipoprotein a is becoming one of the poster children for gene silencing therapeutics, along with PCSK9. I've chosen Lp(a) because we have a drug development program in Lp(a) reduction, so it's easier for me to talk about. Before I go on to that, any questions for now? Okay, let's look at Lp(a). So Lp(a) is a lipoprotein particle that looks a lot like LDL, which many of you will be familiar with, the sort of old-fashioned bad cholesterol. It has an LDL particle at its center, so a load of cholesterol and this apolipoprotein B, the blue wobbly line. The key feature that separates it from LDL cholesterol is the presence of this other apolipoprotein called apolipoprotein a, which is important to distinguish from apolipoprotein big A or big A1, very different. This is apolipoprotein a, which is a protein encoded by a gene called LPA, which we'll come back to. And the Lp(a) particle, which is the particle in aggregate, so the LDL light bit and apolipoprotein a, has effects that initiate and drive atherosclerosis, so formation of plaques in arterial walls, which can then go on to rupture and cause myocardial infarction, stroke, ischemic limbs, etc. It also has a prothrombotic effect, so working in tandem with that drive for atherosclerosis. It makes your blood more likely to clot, and therefore, if the plaque does rupture, then the thrombus, the clot that's formed, is more likely to cause you trouble and lead to an MI or other ischemic event. So it's a nasty lipoprotein, and unusual among the lipoproteins in having this twin effect. It's also quite unusual among the lipoproteins in its population distribution. So LDL forms quite a nice Gaussian distribution in the population. Lp(a), on the other hand, is a heavily skewed distribution, with the vast majority of people, certainly of European ancestry, walking around with little or even no circulating levels of Lp(a). And you have this long right-hand tail of people with high levels of Lp(a). And there's a slightly arbitrary definition of elevated Lp(a), which is people in the upper quintile, which on this scale, in data from the Copenhagen General Population Study, is 50 milligrams per deciliter. Milligrams per deciliter have become old money for Lp(a). New money is nanomoles per liter, because it better reflects the differences in particle size of the Lp(a). 50 milligrams per deciliter is about 125 nanomoles per liter. So mostly I'll be talking today about nanomoles per liter. So we've got this unusual skewed population. An important thing to note is, though, that because there are so many people walking around with little or no Lp(a), and those people have few, if any, adverse consequences of those low levels, lowering Lp(a) pharmacologically is not anticipated to have any major adverse effects. And there are some emerging data that suggest a relationship between low Lp(a) and higher risk of type 2 diabetes, which is not unexpected given the relationship we know about between lower LDL and higher type 2 diabetes risk. But in general, the risks of having low Lp(a) are modest. So why do we want to lower it at all? The plot on the right is data from shows data from UK Biobank. So these are analyses done by a group in Boston, and they show this quite neat log linear relationship between Lp(a) and the hazard ratio for cardiovascular disease events. So this sort of relationship is very similar to that for LDL. But importantly, Lp(a), its association with cardiovascular risk is independent of the association with LDL, also independent of lifestyle, diet, blood pressure, diabetes, all the other conventional established risk factors. Lp(a) seems to stand out. So that's all quite neat looking observational epidemiology, but what about the genetics? And this is where I think it gets really interesting. So this is sort of ancient history data. Now, this is one of the really early genome-wide association studies for coronary heart disease risk, published in Nature Genetics in 2011 by Heribert Schunkert and a huge group from Cardiogram. And you'll see there that LPA, which you'll remember is the gene that encodes Lp(a), or the apolipoprotein (a), is one of the big hits in this GWAS. And more recent GWASs have reinforced this signal and have shown signals with a number of other manifestations of cardiovascular disease, including aortic stenosis, stroke, atrial fibrillation, heart failure, and several others. So there's clearly something going on. When you do the Mendelian randomization studies, you see that a pretty impressive positive causal relationship is supported with atherosclerotic disease, with valvular heart disease, most notably aortic valve disease, ischemic stroke. And this is all borne out by what we know about the molecular genetics of Lp(a), where there are a small number of very well described variants at the LPA locus that determine the size of the LPA isoform, which in turn determines how much Lp(a) you have circulating and how big those particles are. The smaller the particles, the more of them there are, and the smaller the particles, the more damage they do to the inside of your blood vessels, to your aortic valve and elsewhere. So it's a really neat mechanistic link that underpins that G-Well signal. So we're developing an siRNA drug that inhibits expression of the LPA gene, and the intent there is to reduce circulating levels of Lp(a). It uses that same mechanism that I described right at the beginning. And the potential benefits of lowering Lp(a), as you might expect from the plot we just saw from the Mendelian randomization analysis, go across a number of manifestations of cardiovascular disease. Now, the genetic support for this benefit is pretty strong. I know, Stephen, you've been involved in estimating that, which has been really valuable. What we haven't yet got is clinical trial evidence of risk reduction. So there's a large outcome study ongoing with a different type of gene silencing drug called pelacarsen, which is an antisense oligonucleotide, and that's a very large outcome study that should read out in a couple of years' time. And that will be the first really killer test of does LPA lowering reduce risk of cardiovascular disease events. Then there are another couple of drugs coming along behind that. There's one called olpasiran, which is being developed by Amgen, and we're developing an siRNA similar to olpasiran, and there are a couple of others as well. So assuming the genetics is borne out, we should see pretty strong clinical benefit with respect to cardiovascular disease risk. So let's have a little look at the molecule that Silence is developing. So these are data from non-human primates, and data that were presented a few years ago in the American Heart Association by one of my colleagues. I won't dwell too much on them because I think the clinical data are most interesting. The plot on the left shows the plasma concentration of the drug after single administration in monkeys. So this is the pharmacokinetics. I show this because it's interesting in its difference, because of its difference with from small molecule pharmacokinetics. So you see this rapid peak of concentration in the plasma after about 3 or 4 hours, and this is on a log scale, and then there's a rapid decline, so that the drug is almost entirely cleared from the plasma by 24 hours. But you'll remember that the pharmacological effect, the effect on Lp(a), is can be much longer, and we see that again in these same monkeys, that when after a single dose, despite the drug being cleared from the plasma, it's still hanging around and doing its job in the hepatocyte for several weeks afterwards. So this is 3 different doses and control saline, and you see this very long reduction in Lp(a) out to 64 days, and in fact beyond. So there's good evidence this is doing what we expect it to do in animal studies. There's also good evidence that it's not doing potentially unwanted things in animal studies. So I mentioned off-target effects at the beginning. We looked at some specific hypothetical off-target genes in the monkey to see whether administration of this drug reduced expression of those genes in the liver. And the answer was no, we didn't see any effect of those on those hypothetical off-targets. We also see borne out what I was saying at the beginning about the very specific targeting of the drugs to the liver. So this shows data from rats about the concentration of the drug after administration in different tissues. So we've got kidney, liver, lung, spleen, heart, testes, ovary, uterus, and bone marrow. And you'll see, with the exception of liver and kidney, there's effectively nothing to be detected. The big peak in kidney is because the kidney is the primary organ of excretion. So a lot of the drug after being administered goes into the circulation, is filtered by the kidney, and is excreted in the urine. And hence, when you look at the the kidney post-mortem in the rat, you see quite a lot of drug there. The other big peak, as you'd expect, is in the liver, because of that GalNAc ASGPR mediated specific uptake in the hepatocyte. So those are the animal studies that gave us confidence to move into a first-in-human study, which is what I've been working on at Science for the last couple of years. So this was a relatively small study, so only 32 people enrolled. It had 4 cohorts at 4 different dose levels of this drug called SLN360, which is the code name for the drug. So we looked at doses of 30, 100, 300, and 600 milligrams of the drug. And in each cohort of 8, 6 were on SLN360 and 2 were on placebo. So we were then able to pool the placebo group and compare the pooled placebo to each of those 4 dose groups. The people in the study were adults, all over 18, all healthy, so no evidence of cardiovascular disease, but they all had elevated Lp(a), so over 150 nanomoles per liter, which puts them just above the lower bound of the upper quintile in that Copenhagen distribution. So we administered just a single dose to all of these people and followed them up for 150 days, measuring their Lp(a) as we went, collecting information about safety, measuring PK, so the level of SLN360 drug molecules in the blood. And the results look like this. I'm sorry, I should have made these a little bit bigger, but what you've got on the left is the absolute change in Lp(a), and on the right is the percentage change from baseline in Lp(a). There are 5 lines on each plot. The light gray one at the top is the placebo group, and then this brownish is 30 milligrams, the dark blue is 100, light blue is 300, and the yellow is 600 milligrams. The dose was administered here at baseline, and we followed them up for 150 days. And you can see, if we look at the percentage change, which shows it slightly more clearly, that the placebo group barely changed, although there's a reasonable amount of variation around the mean for the placebo group. And then we get this more or less dose proportional effect on Lp(a), so lowering a bit with 30 milligrams and then a bit more with each of the next dosing increments. But the real differentiator seems to be in the duration of that effect. So remember, this is a single dose given here at day 1. Even at day 150, so this is 5 months later, we've got at least 70% lower than baseline in the 2 highest dose groups. And if you remember those plots I showed for inclisiran and the other approved drugs, this is a kind of a familiar looking profile of pharmacological effect. So we're now finishing off this phase 1 study and starting work on a phase 2 study, which should open later this year. And I think even so far, the results on Lp(a) changes are bearing out what the genetics predict. So I just pause there for a moment before we move on to how we look for targets. Any questions? No? Okay. So I said at the beginning that my role at Silence has got two bits to it. The first is working on trials like the one I've just shown you, and the other is working with a group which we call Translational Genomics on the application of genetics and genomics for optimizing our R&D activities at Silence. And that starts with identifying and prioritizing the best targets for our siRNA drugs. We also have a group using machine learning to optimize the design of those siRNA molecules, those 19 base long sequences. And we're also starting to use genetics for stratifying and increasing the precision of our patient populations for trial enrollment. What I'm going to focus on today is the identification of targets. And this all, as you might expect, boils down to trying to understand causal relationships. And this just reflects what that schematic I showed earlier on about this causal relationship flow from G genetic variants through to E gene expression of that gene in a given tissue. And the question of tissue is important. I want to come back to that. And then through to P, the phenotype or the indication, the thing we're trying to treat. In the case of Lp(a), it's quite straightforward, and I think it's slightly unusual in being as straightforward as it is. We've got the Lpa gene, well-characterized variants in the gene with clear effects on the protein product of that gene, not necessarily affected through a classical eQTL mechanism. It's more of a copy number mechanism, but then with a clear path on to the phenotype, which the most proximal phenotype being measured Lp(a) in the blood, but further downstream cardiovascular events. So what we're doing is looking for other targets that are as good as, or potentially good as Lpa for modulation with an siRNA drug. So we can do that in two ways. So framing the question in two ways. The first is starting with the gene and saying, I've got this gene that I think is likely to be a good target. This is my target hypothesis, but I don't know exactly what indications it's going to be useful to treat. What phenotypes does this gene have a causal relationship with? And that's not an uncommon scenario for us, where one of our colleagues in drug discoveries who tend to be biologists, wet lab biologists, come with a target hypothesis and say, what do you think? What does the genetics tell us about how valuable this gene might be for treating a phenotype? What phenotypes could those be? So this is a little bit like almost a PheWAS problem. So you start with the gene and ask, what are the phenotypes that it influences? The other way of framing that, which is probably the most common way we work, is starting with the phenotype. So saying we have an interest in treating, what should we say? Let's coronary heart disease because it's easy. What are the genes that play a causal role in increasing risk of coronary heart disease and have that effect through modulating expression, so through an eQTL mechanism? So this is more of a GWAS, TWAS type problem. And what we're doing to address these, I'm not sure if Dan Wright is on the call because he can give us a lot more color for this. He is, I think. Is using a combination of co-localization and Mendelian randomization. So the reason for doing this is that we want to give ourselves the greatest possible confidence that the gene we think is having the effect on the phenotype is in fact that gene. So confirming the signal to gene mapping, because if we get the gene wrong, then everything else downstream is sunk. And also that confirmed gene is having its effect on the phenotype through expression. So what we're doing is co-localizing a GWAS signal for the GP relationship with an eQTL signal for the GE relationship. And if we get a positive co-localization signal, that's the first step in our exploration. You can do that at large scale across the genome and across the transcriptome. The question then is, yes, there's a positive co-localization, but are the signals in the right direction? So what we want is a situation where lowering the expression of the gene, so of transcript E1, has a beneficial effect on P, because our drugs downregulate expression. So it doesn't serve us well to have a gene where upregulation of expression is required in order to have the beneficial effect. That doesn't completely eliminate that type of scenario from feasibility. In fact, we do have our other major clinical program uses a negative regulator as its target. So it downregulates a negative regulator of hepcidin in order to increase hepcidin. So our goal is increasing hepcidin. Our drugs clearly can't directly increase hepcidin, but by downregulating a negative regulator of hepcidin, we get an increase in hepcidin. So it's possible to handle those, but our first pass is to look at genes where downregulation appears to have a beneficial effect on the phenotype. We can then superimpose MR methods on top of that and confirm the direction and possibly the magnitude of that causal relationship. Dan, is there anything you want to add to that?
Hello, I'm not sure how good my microphone is. Can you hear me?
Yes, a little bit quiet, but we can hear you.
Yeah, sorry. No, nothing in particular. Yeah, really good summary. I don't think there's anything for me to add. Thanks.
So that all sounds quite rosy, but I've got some questions. I really value your thoughts on those in a moment, but any questions from others before I move on?
Can I ask a question?
Yeah.
Yeah. Sorry. Xin Yuan from the Stats Lab. So I'm just wondering why we need to identify the genes instead of would it be sufficient to just identify the expression, the gene expression that's useful in this pathway?
Yes. Good question. So you're right that it's the transcript, it's the expressed mRNA that is the really critical mediator of this whole thing and is the target of the drug. But we need to go back to the gene so that we can make sure that the causal pathway is anchored at the DNA end. So if you just looked at the gene expression phenotype relationship without considering genetic variation, it's possible that observed differences in gene expression may be a consequence of the phenotype rather than a cause of the phenotype. So introducing G anchors the causal pathway, and you work on the assumption that the causal effect flows from G to E to P.
Okay, thank you.
I see Stephen Burgess, you're on. You can certainly cover that a lot more eloquently than I can. So these are some conundra that I wanted to bring up, because I think these are probably things that you've given some thought to. There are some cases where it's really easy, and there's a single gene at a locus where we observe a signal, and it's clear that that single gene is the most likely culprit for the signal, and we can ascribe the downstream causal effects to that one gene. There are also an awful lot of circumstances where we see a crowded locus, where there's more than one gene, and often more than three. And distinguishing those from each other is not always straightforward, even with good quality EQTL data. So this is something that we're still working on, but I would be keen to hear thoughts of those who've been thinking about this for a while on how we might address this sort of problem, because it's often the locus with many genes in it that looks most promising, but we just can't pin down exactly which is the gene that's driving it. Any thoughts on that?
Yeah, Dan, I might chip in, Steve Burgess.
Oh, hi, hi, Steve.
Hi, thanks. Really interesting. Really, yeah, really appreciated Xinguan's question as well. We've been trying to work on this in kind of a multivariable Mendelian randomization setup. I'll share a link to a draft paper in the chat in a moment. But
Thanks.
yeah, trying to so one of the difficulties is almost a kind of Goldilocks problem. If you include too few variants in such an analysis, then you can have fewer independent genetic predictors than you have phenotypes. So you're essentially trying to estimate more parameters than you have data points. But if you include too many variants in the analysis, you can get kind of almost over prediction. Yeah, almost the model is just very, very tight, and you end up being very sensitive to estimates of variance correlation matrix. We've kind of approached this trying to use a dimension reduction approach. So trying to reduce the genetic variance to a series of principal components, such that they explain a large proportion of variability, but without being highly correlated.
Yes.
So that's one kind of idea that we've had. At the moment, we've looked at it with protein data rather than gene expression data. But I don't see why it couldn't be used with gene expression data as well.
No, should be pretty similar. That's really neat. Yeah, thanks. Although my second question possibly makes it more complicated than the protein data, because I guess plasma proteins are measured in one tissue in plasma. You know where they are, you know what they are, and it's pretty straightforward. Gene expression data, so something like GTEx, where you've got gene expression measured in dozens of tissues, often throws out results where you've got positive EQTLs in multiple tissues, which means you can have positive co-localization in multiple tissues. So say tissue 1 here is the liver, which is great. We like genes that are expressed in the liver because that's where we can act, but you might also see expression in kidney and in prefrontal cortex, and you get positive co-localization with each of those 3. A question we often get from our biological colleagues is, well, that's nice, you've got a positive signal in the liver and it co-localizes beautifully, but how do we know that the true causal effect is not being driven primarily by one of the other 2 tissues or both the other 2 tissues, and the liver effect is secondary or less important than those? And also, I suppose, does it matter that we're knocking down, does that make sense? No, forget that. So the question is really about which is the tissue that's really having the biological effect, or is it more than one, and does it matter, and how do we dissect those apart?
Hi, Dan, this is Chris Wallace.
Hi, Chris. Hello.
I don't think you can dissect that apart with observational data, can you? mean, if it's exactly the same transcript in all the tissues, then you're going to need to actually intervene at some point, right?
Yes. Yeah. I think that's sort of where we're coming to. So I suppose the next question is then, does it matter?
Probably depends on the disease.
Yeah.
I mean, if it's Alzheimer's and one of your things is in the brain, then maybe.
Yes.
If it's gout, then maybe not.
But say it is Alzheimer's and you see positive, and your drug is liver targeted, and you see positive signals in liver and in brain, how valid an assumption would it be that downregulating the gene in the liver, because you can access it, may have a beneficial effect in the brain? Is it possible to work that one out?
I think you've got to look at an animal model for that, haven't you?
Yeah.
Is there potential here, Dan, if you have sufficient tissue-specific EQTLs for the expression in each of those 3 tissues to model it in a multivariable MR with 3 separate instruments, where the instruments are tissue-specific? I'm just thinking aloud. All the experts are on the call, so please correct me.
But if they're all co-localizing, then the instruments can't be tissue specific, right?
Right.
And that the instrument is shared.
Yes. Yes, indeed.
So then, if I flip my previous question around, is the presence of a positive co-localization with a tissue, an expression in a tissue, evidence that that tissue is playing some role, even if it's not the dominant role?
Not in my view.
No.
It's not sufficient. It's necessary, but not sufficient.
That's interesting. Yeah, and that sounds very much like the debate we've been having internally.
And can I throw something else in the mix? Does it matter that all of our expression data comes from healthy controls?
Well, yeah, it probably does. And also, I mean, GTEx, they're healthy-ish. Most of them are either nearly dead or recently died. So yes, they don't have a specific diagnostic label, but they're not in rude health. But even where we do have, in other EQTL data sets where they're in healthies, I think you're right. We're probably missing some disease-specific effects, and it's for that reason that we've gone out and started generating our own disease-specific, tissue-specific EQTL data.
Dan, I'm going to interrupt here just because I'm conscious of time.
Yes.
And I suspect there's a few of us on this call that could go on to have a very long conversation. But because it's almost 3 o'clock, I wanted to thank you for a good talk. I'm not closing the conversation.
That's all right.
But I wanted to thank you for talk and ask if there's anyone else in the audience who needs to get through who wants to ask a question of you. Raise your hand now or forever hold your peace. Okay. Perhaps not. So I'm happy to stay on and have a little chat if other people are available to.
I've got to zip off, I'm
You've
afraid.
got to zip. Okay.
But Dan might, if you're up for it and free, that might be great actually, if you're keen.
Yeah, no worries. Yeah, that would be good.
Yeah, thank you. And sorry that I've got to go to a meeting with our board, which I probably shouldn't be too late to.
Okay, I wanted to ask one other non-co-localization question.
Yes.
That all this sRNA work about degrading the expression of a gene, and you did mention that sometimes there are negative regulators that you could degrade instead. But if you need to raise expression of a gene, how often is it that there are these regulators you can target? Or how often is it that actually what you want to do is degrade the gene? You know, how often is it? What's the balance?
Generally, I think turning things down is more often what you want to do than turning things up, which is good because the technology for turning things down is better established. There are small activating RNA drugs being developed that have the opposite effect. They try and increase expression of a given transcript. It's a much less developed technology, but it's happening. I only know of one target, although there are probably others, where the target is a negative regulator that seeks to upregulate something, which is this TMPRSS6, which we're working on, and a couple of other companies are also working on, which is the hepcidin example. But it's something we've thought quite a lot about, where we need to turn something up, but we can only turn a gene down. Can we use pathways or some other sort of serial MRs to look at different steps in a pathway and find the closest negative regulator to our intended target? I think that could be really interesting. That's where sort of graph-based methods come in.
Thank you. I'd be interested to hear more about that some other time. But it is 3 o'clock, so I'll say thank you again and let you run if you have to run.
Well, thank you. Thanks ever so much. Really good to see everyone. Thank you for a great conversation, and thanks for humouring me with chucking some questions back over the fence at you. But I better run off, but I will leave you with Dan, and I look forward to speaking again soon.
Thank you.
Thanks, you too. Bye.
So I don't know, Dan Wright, Steve Burgess, if you want to hang around and chat. I've got maybe 15 minutes, and anyone else is welcome, of course.
Yes, I'd love to. I'm not sure I've got I would love to have a chat with both of you about all of these methods in a lot of detail. I haven't particularly got anything structured to ask you in terms of any specific questions.